Okay, everyone, let's move on now into the next chapter. So our next chapter here for microbiology is going to be on microbial metabolism. Remember, we've kind of started a section of the course where we're looking at microbial physiology and how microbes work and what do they do. So last time we looked at microbial growth and nutrition, and we'll delve a little bit deeper now in chapter 7 and look at microbial metabolism. Okay, now let's get started. So first, a little bit of history. It's always important to give credit to scientists and people who made these groundbreaking discoveries. Louis Pasteur, pictured here and here, was a French biologist who worked in the mid-1800s and is a real key figure in that golden age of microbiology we talked about. Now, one of the things that Louis Pasteur did early on, 1857, he was able to show that if you heat a mixture to a hot enough temperature for a long enough time, you will kill the microbes, the yeast that's in that solution, and by killing the microbes, you stop fermentation. So he was able to show that it's the microbes that are causing the fermentation. It's not that the fermentation attracts microbes. The microbes are the cause, the source of the fermentation. So this event marks the start of that golden age of microbiology. In 1864, he would develop of course, even we use it today, the very famous technique pasteurization, named after Louis Pasteur. Pasteurization is a technique used to control the growth of unwanted microorganisms in food. Pasteur was working with wine. By applying pasteurization to the wine, he was able to prevent premature spoilage, and it really saved the wine industry at the time from disaster. So, metabolism. What do we mean by metabolism? Metabolism is all of the chemical reactions that occur in a cell, and it refers to an organism's ability to use and create molecules to sustain life. The ultimate goal of metabolism is to make sure that an organism can reproduce. There are many effects of metabolism, everything from causing bread to rise. The CO2 is released by the yeast, to botulism, to bad smells, etc. And we can perform tests to check for metabolism or the byproducts of metabolism. Okay? Now, metabolism can roughly be broken down into two general categories. First, we have anabolism. Anabolism are reactions that build larger molecules from smaller molecules. So if we take a whole bunch of small molecules, which earlier we talked about are called monomers, and we hook them together to make something larger and more complex, we call that anabolism. Anabolism is used, it's going to use energy. We need energy to build that bigger molecule. And once we have a big molecule, that represents potential energy that can be used later if we break it down. When we perform anabolism, when we build large molecules from small molecules, it often involves what are called reduction reactions. Reduction means a molecule gains electrons. That's how we abbreviate electrons. But if a molecule picks up or gains an electron, it's reduced. That's a reduction reaction. Now, the opposite is catabolism. Catabolic reactions, or catabolism, these are reactions that break down larger molecules into smaller molecules. When you do that, you release energy. We break something down. Now, catabolism often involves oxidation reactions. Oxidation is when a molecule loses an electron. So you can remember the saying, Leo goes grr. L-E-O, like Leo the lion. Leo, loss of electrons, is reduction. Okay? Gain of electrons is reduction. Loss of electrons is oxidation. So Leo, loss, electrons, oxidation. Grr, G-E-R, gain, electrons, reduction. So Leo the lion goes grr. Okay? So we see here, this atom here has lost an electron, so it is oxidized. This one has gained an electron. It picked it up from that one, so it is reduced. Okay, Leo the lion goes grr. Now, in many cellular oxidation reduction reactions, which we shorten to redox, oxidation reduction, in many cellular redox reactions, electrons and hydrogen atoms, abbreviated here, are removed at the same time. So as we see here, going along with that loss of an electron, we're also losing a hydrogen. And something will pick up a hydrogen and pick up an electron. So oxidation and reduction. Now, we can put all that together in what's called the ADP, ATP, energy carrier system. Now, as we've mentioned before, in living organisms, in cells, energy, which we're going to abbreviate here as a capital E, energy is stored as an organic molecule called ATP. ATP, pictured here, stands for adenosine triphosphate. So we have this adenosine piece of it and 1, 2, 3 phosphorus atoms attached to it. So adenosine triphosphate. This represents chemical energy, potential energy that can be used in living systems. So how this works is cells break down ATP when they need energy. So we're going to take our ATP, we're going to add some water to it, and that's going to break our ATP down. What we are going to do is remove this third phosphorus. That leaves us with ADP, adenosine diphosphate, only 2 phosphates. Here's the phosphorus we removed, but since we started complex and we broke it apart, we kick out energy. All right, so that releases energy. So when cells need energy, we simply have to break down ATP. Because we added water to ATP, this is called a hydrolysis reaction, and this would be an example of what we just talked about, catabolism. We're breaking something down to release energy. If cells want to store energy, they can make ATP by doing the reverse reaction. We can go this way, right to left. So we take our ADP here, add a phosphorus to it. Now, to add that, to build that, we have to kick in energy. And that's going to give us ATP, and we're going to kick out water as a waste, so this is called a dehydration reaction. This would be an example of anabolism, building something up. So ATP connects anabolism and catabolism. Cells are constantly making and breaking ATP. We're making ATP to store energy, and we're breaking ATP to release energy. So we're constantly making and breaking ATP. So looking at that a little bit further, how can we produce and then store ATP? There are several ways to do that. One way that cells often use is something called substrate level phosphorylation. Now I know that's a mouthful, but all we're doing here is we're pulling a phosphorus off of another molecule and then using an enzyme to add it to ADP. So I'm going to go up to some other molecule that has a phosphorus. We're going to steal that phosphorus from it with an enzyme. I'm going to add that, bond that to ADP, and it gives me ATP. You need hydrogen to power that reaction, but you do not need oxygen. So it's pictured here. Here's our molecule here. It's got a phosphorus. This big enzyme is going to come along. It's going to steal that phosphorus, add it to ADP, and give us ATP. Okay, we're also picturing that here. All right. Another way to produce or store ATP is photophosphorylation. Now, photo always refers to photosynthesis, so we're going to be talking about that here. Photophosphorylation is used by photosynthetic cells. Light energy is used to make ATP from ADP. This does require oxygen, and it's going to use light energy, and we're going to make ATP from ADP. Okay? Now, it involves a complex series of reactions, as illustrated here, more than you have to know. But what you do want to know is photophosphorylation is going to make ATP from ADP using light energy. A third way is called oxidative phosphorylation, and this can give us the most energy. Now, what this refers to is where we're going to make ATP through the use of an electron transport system. Oxygen is necessary, but you can generate large quantities of ATP. Now, all we mean by an electron transport system is we're going to take an electron and we're going to hand it off to a molecule. And then that molecule is going to hand it off to another molecule. And that molecule is going to hand it off to another molecule on down the chain. So we can see here, here's my initial electron, and we're going to bounce it down this chain from molecule to molecule to molecule. Every time we transfer an electron from one molecule to another, it loses a little bit of its energy, and that energy can be used to make ATP. This is called oxidative phosphorylation. Here we see it in reality. We know what the molecules are that these bounce around to. Notice the yellow and the orange arrows here. Look at all the oxidation reduction going on. Anabolism and catabolism. And we're going to make a large quantity of ATP. Now, everyone, all these reactions require enzymes. As we talked about a little bit earlier, enzymes are globular proteins. They have kind of a globular blob-like shape. They're globular proteins that act as a catalyst in living systems. Now, a catalyst is something that speeds up reactions. So all enzymes are proteins. Not all proteins are enzymes. So what are some characteristics of enzymes? Well, again, all of them are globular proteins, not straight line, but sort of blob-like. They all cause reactions to occur faster, and they are extremely good at what they do, extremely efficient, effective even in low numbers and low concentration. For example, the turnover rate or turnover numbers is anywhere between 1 to 10,000 molecules per second. So the enzyme is making 1 all the way up to 10,000 molecules of what it wants to make per second. What this translates to is the enzyme causes reactions to occur millions or billions of times faster than normal. Now, to do that, enzymes have to be very efficient. They work on only one specific substance, which we call a substrate, and for only one particular reaction. So one enzyme to one reaction. There aren't really generic enzymes that work on more than one. And by definition, catalysts, and this would include enzymes then, catalysts are not used up in their reactions. They're not part of the reaction. They just make the reaction occur faster. So they can be used over and over again, making them extremely efficient. They are also extremely important. Life would not exist without enzymes. because the biochemical reactions that we need to sustain life would occur far too slow without enzymes. Now, just a quick note on naming enzymes. Not all, but most enzymes end in the suffix ase. So something like sucrase would be an enzyme. Now, how do enzymes work? Enzymes work by lowering what's called the activation energy of reactions. The activation energy is the energy needed to get a reaction started. So if we look at this diagram here, here's my starting material, my reactants. That's my starting material. I want to split this apart, and here are my products. Now, to break that chemical bond there, it requires all this energy. This wall here in gray represents activation energy. So to break this apart, we have to kick in enough energy to go over that wall. This is without an enzyme. Look what happens if I use an enzyme. You see how we've shortened that wall? Now it's just real easy. So the enzyme reduces the activation energy, lowers the activation energy, and that makes the reaction occur faster. We can see that in a graph form here. Now, how an enzyme does that is enzymes bind with their substrate. Remember, the substrate is the one substance the enzyme works on, and it increases the likelihood of a reaction occurring. When an enzyme binds to its substrate, it does so at what's called the active site. All enzymes on the enzyme, there's an active site that binds with the substrate. When the enzyme and the substrate are together, we call this the enzyme-substrate complex. And this binding between substrate and active site is very specific. It's similar to like pieces of a puzzle fitting together or a key fitting into a lock. Not exactly lock and key, because the active site can change its shape a little bit, just slightly, to accommodate the substrate. We call this the induced fit model. But the binding is very, very specific. Keep in mind that enzymes can both degrade, break down things, and build things up, synthesize. So we can see here. Starting at the bottom, here are my substrates, my materials. What I want to do is chemically hook these together to make this. Here's the active site. You will notice that these substrates fit in there, lock and key. Here's my complex. There's my product. Enzyme lowers activation energy, and this enzyme is not used up. It can be kicked out and continue working. Here we see this is a synthesis. Here's degradation, breaking down. Now, being such important molecules, enzymes do not work all by themselves. They have coenzymes. You can think of a coenzyme as an assistant. It is a separate organic molecule, carbon-containing, that assists full enzymes. In living things, vitamins act as coenzymes. There are also cofactors. This is another helper molecule, but it's inorganic, does not contain carbon, and it's not a separate molecule. It's actually part of the enzyme, and it gives the active site its proper shape. Now, in living things, minerals are cofactors. So we see here, here's our enzyme, there's the cofactor in red, and that gives this active site its proper shape so that the substrate can fit in there, lock and key. Now, environmental conditions can also influence enzyme activity. If we change the temperature or pH, you will greatly affect how the enzyme works. This can both inhibit, slow down, or speed up enzyme activity. If the temperature gets too high, or the pH gets too high or too low, enzymes will denature. they lose their structure and kind of fall apart and no longer function. If an enzyme gets too cold, it doesn't denature, but it slows down. So you can see here, pepsin here is an enzyme in your stomach. Notice it works best at low pH because your stomach's acidic. It's pH 2. Trypsin's in your small intestine. It wants to be more of a neutral pH, around pH 7.5 to 8. Now those numbers aren't important, everyone. What's important is the shape of this curve. Look how quickly enzyme activity drops off if the pH gets too low or too high. It's like falling off a cliff. And the same thing with temperature. This enzyme works best about right here. If that temperature gets too cold, enzyme activity slows down gradually. If it gets too hot, it denatures and quickly stops functioning. Okay? So here would be an example of a functional protein. Here's one that's denatured and it would no longer function. So, besides changing temperature and pH, another way to affect enzyme activity is through something called feedback inhibition. Now, this is very important. This is where the end product of a reaction inhibits or stops the reaction itself. Okay? This is how your furnace works, for example. What's the end product of your furnace? Heat. When enough heat builds up and it hits the temperature you set your thermostat at, what happens? Your thermostat shuts off. So the end product, the heat, shut the furnace off, the very thing that was making it. Okay? In living systems, feedback inhibition is very important and might work like this. Now, here's my starting material, A. My end product is D. I'm going to have to make B and C to get to D. Each one uses a different enzyme. When enough of D builds up, D can feed back and shut down that first enzyme. And notice if I shut down this first enzyme, the whole thing is shut down. So in living systems, many, many reactions are controlled by feedback inhibition. Now, all of this, everyone, can come together in what's called a metabolic pathway. This would be a series or sequence of enzymatically catalyzed reactions in the cell, where the product of one reaction becomes the starting materials or the reactant of the next, and so on. So we might see it like this. Here's my starting material, A. I want to make E. To make E, I have to make B, C, and D. So this is one pathway. A to E, I might start with C, and another pathway could be C to G, but I have to make F first. Notice that they each use their own enzyme. Now, these things made along the way. Now, what I want to make here in this first pathway is E. But to make E from A, I have to make B, C, and D first. This isn't what I want, but they're necessary steps along the way. B, C, and D are called secondary metabolites or metabolic intermediates. Same thing. These are substances formed in the process of going from reactant to product. They're not essential for routine survival, but they can provide some side benefits. For example, naturally produced antibiotics and some pigments, like this color we see in this bacteria, are secondary metabolites. Secondary metabolites can also be what are called exotoxins. Now, exotoxins are highly potent toxins that are secreted by some bacteria. Now, you might remember in the earlier chapter on prokaryotes, I said gram-negative bacteria have endotoxin. Endotoxins are only released if the cell is damaged or disrupted. So they're not secreted, they're not purposely released. Exotoxins can be purposely released by some bacteria, and they damage host cells and disrupt the host cell's metabolism. Cholera, diphtheria, tetanus, all examples of exotoxin-producing bacteria. And remember, these exotoxins are not made deliberately. They're simply intermediates, secondary metabolites produced along the way. Now, as a good example of everything we just talked about, we're going to take a brief look at carbohydrate catabolism, breaking down carbohydrates. Carbohydrate catabolism is central. It's the most basic type of metabolism, and it's a great example of how metabolism works. Now, in microbes, we can break down carbohydrates a few ways. We can do it with oxygen. That's called aerobic. This is called cellular respiration. We can do it without oxygen, and this can be what's called anaerobic respiration or fermentation. So metabolism as a whole is outlined here. Make sure you know this for the test. No, just kidding, everyone. This is just to show you how complex metabolism can be inside a cell. So breaking down carbohydrates, if I use oxygen, that's cell respiration. If I don't use oxygen, I can use anaerobic respiration or fermentation. Let's start with cell respiration. So say we're going to use oxygen. Now, cellular respiration is this process. I'm going to take glucose, a sugar, and through a bunch of reactions here, I'm going to convert that into usable ATP. Remember, ATP is energy, chemical energy. We're going to kick out carbon dioxide and water as a waste product. So this is the equation. I'm going to have my glucose. Glucose is C6H12O6. We're going to add oxygen, and we're going to start with 38 ADP. From that, I'm going to make 38 ATP, energy, but I'm going to release water, and I'm going to release carbon dioxide. In eukaryotes, you can only make 36, just for completeness. Now, this diagram may not make a lot of sense now, but it will when we're done with this. Basically, here's our glucose that we're starting with. Now, in step 1, to break apart glucose in cell respiration, we're going to do something called glycolysis. Glycolysis is where we're going to convert our glucose into something called pyruvic acid, which is a 3-carbon molecule. So here's my glucose, everyone. Through a series of reactions, we're going to convert that to right here, pyruvic acid. That's glycolysis. We're going to use enzymes here, of course, and we're going to produce some ATP from that. Look at here, glycolysis, I get a little ATP from that. And I also get a little of this molecule called NADH. NADH represents not real, but temporary energy. It's a molecule that temporarily holds on to electrons. So think of this NADH as temporary energy. Okay? So I'm going to take my glucose, I'm going to make my pyruvic acid. We're going to kick out some real energy and some temporary energy. Now, if we have oxygen, this is aerobic, I'm going to go this way. Okay? So now I'm going to take my pyruvic acid. This is actually glycolysis if you're interested. I'm going to take my pyruvic acid and we're going to do something called the preparatory reaction. So I'm going to take my pyruvic acid and I'm going to break it into a 1-carbon piece. Remember, it's 3 carbons, and a 2-carbon piece. The 1-carbon piece combines with oxygen, it becomes CO2, and it's released as a waste. You're breathing it out right now as a waste. The 2-carbon piece is picked up by a coenzyme called coenzyme A, and together it's called acetyl-CoA. Now, all this acetyl-CoA does is it's going to transition to the next step. It's sort of a preparatory reaction or transitional step. What acetyl-CoA goes into next is the third part called Krebs cycle. Krebs cycle, there is still a lot of energy in this 2-carbon fragment, in this acetyl-CoA. So a great amount of energy remains in the chemical bonds of acetyl-CoA because there's a lot of electrons there being held onto. Now what happens is these electrons that are in acetyl-CoA are transferred to two different coenzymes, NAD and FAD. So through a series of reactions, we're going to take the electrons that are in acetyl-CoA here, and we're going to hand them off to NAD or FAD. Remember, these coenzymes are temporarily holding on to electrons, so they represent temporary energy. To put this in perspective for you, NAD is also called niacin or vitamin B3. And FAD is also known as riboflavin or vitamin B2. Now, during Krebs cycle, when we hand off these electrons to NAD or FAD, we're going to produce carbon dioxide as a waste, and Krebs cycle occurs in what's called the cytosol in prokaryotes. The cytosol is the liquid portion of the cytoplasm inside the cell. In eukaryotes, it occurs inside the liquid portion of the mitochondria, an organelle called the mitochondria. Here's Krebs cycle in depth. If you're interested, you don't have to know all that. And then the last step is the electron transport system. Now remember, after Krebs cycle, we have our coenzymes, NAD and FAD. They're holding on to all this temporary energy. And remember back, we made some temporary energy from glycolysis. So there's some NADH there. That's temporary energy. What we're going to do with the electron transport system now is we're going to generate most of our ATP from catabolism. Now, electron transport occurs in the plasma membrane in prokaryotes, and it occurs inside the mitochondria of eukaryotes. The cristae is the inner membrane of mitochondria. Basically, it is a series of electron carrier molecules that are in turn oxidized and reduced as they get and then lose an electron. So, NAD and FAD are going to transfer their electrons to an electron acceptor molecule. Remember, everyone, in an electron transport system, when we hand off an electron, the electron's going to lose some of its energy. So NAD and FAD give their electrons to an acceptor molecule. That molecule gives it to another acceptor molecule, then that one to another acceptor molecule. So the electrons pass through a series of different molecules. That's called an electron transport system. Remember, they lose some of their energy during each transfer. This lost energy can then be used to form ATP in the reaction we saw earlier, oxidative phosphorylation. The enzyme that catalyzes this reaction is called ATP synthase. Notice the ASE on the end. Now remember, we're bouncing electrons from acceptor molecule to acceptor molecule all the way down that electron transport system. The final molecule in the chain is good old oxygen. At the very end of the electron transport system, oxygen receives 2 hydrogen and 2 now low energy electrons. If I give oxygen 2 hydrogens and some electrons, what's H2O? Those recombine as water, and that's released as a waste. Your body makes this all the time as waste. It's called metabolic water, and you will eliminate that in your urine. Without oxygen, this whole chain backs up and stops, leading to death, sometimes quick death. It's how cyanide works, as it binds with this oxygen, backing up the whole chain. All right, there's our electron transport system in detail, where it occurs in bacteria. So if we go back here quickly, let me run all the way back to this diagram here. All right, we did glycolysis. There's my pyruvic acid. There's my preparatory reaction. Here's my acetyl-CoA. That transfers the Krebs cycle. Now notice as Krebs cycle turns, we're making all this temporary energy, FADH and NADH. We're holding on to these electrons. We transfer those to the electron transport system, and that's going to crank out all our ATP. Okay? All right, so that is an overview of carbohydrate catabolism. Now, I'm going to split this chapter into two parts like I did the last one. Next, we're going to talk about anaerobic respiration and a couple other aspects of metabolism. So this is it for part 1. I'll see you back shortly for part 2 of this chapter. Okay, see you soon.